today we're going to get on to a really central and interesting part of cognitive neuroscience, and something which I started talking about in the last couple of lectures, particularly in vision, where I was talking about the binding problem. We're going to talk about how the higher centres function together to produce the kind of very high level processes that we understand as cognition and awareness and consciousness and thought and all of those things. So the picture, first of all, on the first slide is a still taken from an animation, and it's something that identifies both parts of the binding problem. Okay, what's it a picture of? A woman, okay. I have another question for you. Have a look at it for a few minutes and decide to yourself whether she's standing on her left leg or her right leg. Okay, hang on. Put your hands up if she's standing on her left leg. And put your hands up if she's standing on her right leg. Okay, so we've got nearly half and half. What's happening here is that we haven't got a woman spinning around at all here. What we've got is a series of black splodges of different shapes being animated one after the other. But your visual systems and your cognitive systems are putting that information together and adding extra information to it to make it into an image of something that you're familiar with. And when you add that information in, some of you are adding the information in and making a stand on one leg, and some of you are adding the information in and making a stand on the other leg. And if you look at it for long enough, you can actually make her stop and spin the other way and stand on the other leg as she's doing that. But whenever you're faced with a sensory situation where there isn't enough information to make sense of it, your brain will always add in the extra bits and make sense of it for you, because it's easier than being confused by the world. So that's an illustration, really, of the spatial binding problem. We've got a black splodge and we call it a woman. And the temporal binding problem. All those black splodges are all the same shape in sort of different orientations. And we're building a three-dimensional construct out of a two-dimensional image just to answer the question and make it one structure that moves rather than make it a series of structures. So this is a microphone placed near to somebody who's clapping in a crowd of people who are clapping, applauding at the end of a concert. And you can see that the sound is oscillating up and down, and you can get nice peaks and troughs where that person is clapping. If you put the microphone above the whole crowd, so that you're picking up from everybody, you can see that there are periods where there's just sort of general activity, and then there are some periods where the whole crowd seems to be clapping together in unison. Yeah? Have you all been in a crowd where that's happened? Yeah, you're applauding the band or the play or whatever. Suddenly, for a period of a few seconds, everybody's clapping together and then it all collapses and goes off again. That's, again, an example of an emergent function. Nobody's in charge of that, but order seems to come out of the disorder and then kind of sink back down into the disorder again. What's interesting is that if we look at the energy levels in the sound in the room, generally, we find that when the people are clapping in unison with each other, there's less overall energy than when you've got that kind of discordant clapping. So what I'm saying is, if you've got a system that is functioning in an ordered way, it uses up less energy than a system that's functioning in a disordered way. Whether or not you have anybody in charge of that order or anything controlling that order, the system will tend to order because order uses up less energy than disorder. And so it will just kind of appear out of the nothingness. It's emergent. Here's another example. This is water going down the sink in my lab. Me videoing this, convincing my lab tech that I was absolutely mad. See this flute that comes down the middle here with all those ordered patterns in it? Who's in charge of making that? Is it that water molecule, or is it the piece of metal there in the sink? It's just something that appears out of the disorder of this water draining down the sink. And the reason it appears is because this is the system for the water draining that has the less energy, or is the most efficient compared to all the other systems. And so the system is capable of finding for itself its most efficient setting. And that is where we think the central nervous system is when we talk about oscillations and rhythms within the central nervous system. Neuroscientists have only really begun to consider these oscillations and rhythms over the last 20 years or so. And over the last 20 years, the consideration of these oscillations has been the single most important change in cognitive neuroscience. It's a difficult thing to get your minds around and a difficult thing to sort of work through and understand. But if you're going to understand a modern concept of how a complicated brain works, this is the most important feature to understand. This is the cover of a neurophysiology textbook from the late 80s, about 87, 88, I think. This is Carpenter, and this is the second edition, if I remember rightly. And this is an illustration of the central nervous system. And what we've got here are sensory neurons sending information to progressively more and more complex layers and coming out here in motor neurons. Yeah, so the whole system is moving through. And if you think about it, this is the kind of picture that I was trying to build a couple of lectures ago when I was talking about ganglion cells and simple cells and concentric cells and complex cells, layers and layers of cells, yeah? Building the information, constructing more and more complex interpretations of the sensory information, and the sort of assumption there was that eventually this would all end up in some motor function somewhere. Yeah, the whole thing would tumble out into something that would be a response to the environment. If you look at this diagram and think about it, what it's saying is that the whole of the central nervous system is one big reflex machine. And that the sensory inputs are A, really important, and B, actually the thing that are driving the whole system. If you consider that system without sensory input, you haven't got anything that's functional. You've just got all these weird connections waiting for something to happen at this end. So the whole system is driven by the sensory input. And this is a kind of mechanistic behaviorist view of an animal, which was prevalent, as I say, in the 80s. People were beginning to talk about experience as being due to sensory input and reflex responses in inverted commas. Let's look at a much more modern explanation of the central nervous system. Here we've got a group of neurons in a loop with dendrites here, cell bodies here, and an axon here going to the next dendrite with chemical synapses between them. And you can see that the way this is illustrated, there is information travelling around this loop so that the information is here, before it's here, before it's here, before it's here, before it's here, and then it gets back to here and so on. The information is going round and round in a loop. And this is a much more modern understanding, or concept, I guess, of central nervous system processes. Now, if you look at this model and compare it to the last model, the sensory input is much less important. Yep. What's important to this model is what's going on inside its head. Yeah, all those things, those homeostatic processes and those deep, meaningful thoughts that we all have at 8:15 in the morning are all going on inside. And sure, if there's somebody droning on at the front, well, that's quite important, I guess. I'll pay some attention. But really, it's peripheral. What's really important are all these oscillatory, circulatory processes going round. And that is our current understanding of how the whole system works. And it's turned the importance of sensory input on its head to a point now where people are almost saying, well, if we had a central nervous system with no sensory input, it would be all right, really. It would carry on. Its oscillatory processes would carry on. And it might be a little bit denuded and not as good at things for not having any sensory input, but broadly speaking, it would get on, it would be all right, all the processes would still work. It certainly wouldn't collapse in the way that the older model would have collapsed without sensory input. So that's why these oscillations are so important. They drive the whole system. They ensure that the system is more efficient than it would be if it didn't have oscillations. And anyway, who needs sensory input? It's overrated. It's all about what's going on inside the machine. And yes, the machine can alter what it's doing in accord with its sensory input, but those are not central anymore. All the older concepts have kind of taken a big step backwards, and this oscillation has replaced them. So I guess we need to talk about oscillations and how they fit into CNS processing and how they drive the whole system. First of all, I need to talk about two different kinds of oscillations. First kind you'll have seen if you've ever been in the foyer of the science towers. Everybody seen this thing that goes on forever and nobody can quite work out what it's for, but the physicists think it's really cool? It's an oscillating pendulum. And it goes across and across and across, and anybody that's done physics will tell you that it goes across in exactly the same time each time, and it's really stable and it's great and so on. It's a harmonic oscillator. It's one of the two major kinds of oscillation. And harmonic oscillators are all great, but they have their problems. Has anybody ever crept across the barrier and given this a good kick? No. Apart from the professors in physics descending on you and beating you up big time, what do you think would happen? Would it carry on or would you disturb it? You'd disturb it. And how long would it be disturbed for? Yeah, for ages at least, and probably forever. If you disturb a harmonic oscillator, this is the least bad that you can do. If you disturb it in synchronization with its oscillations, i.e. you go along and you gently give it a tap in the direction it was going anyway, you will alter the amplitude of its oscillations for something like 10 wavelengths or so. So if that thing has a wavelength of, 2 seconds maybe, just by tapping it gently, you're going to alter its function for 20 seconds. You're going to have an effect on its function for a long period of time. And if you should tap it in a different direction to the direction it was going in, then things will get really complicated. Not only will you alter its amplitude for a period of time, but you'll alter its wavelength as well. And it will take, again, up to about 10 wavelengths to settle back down into a rhythm. So harmonic oscillators are interesting, but they're a little bit temperamental. They're all right if they're left alone, but they're not great if you kick them, tickle them, play them. They don't deal with sensory input very well. The other kind of oscillator, but it's well illustrated by this dripping tap, again from my lab, where all the taps drip most of the time. What we've got here is an oscillatory process going on with a remarkable degree of regularity, but it's just pulsing every so often. It's building up, building up, building up, and pulsing. And then building up, building up, building up again, and pulsing. What would happen if you flicked that tap just about the point that this drop is about to release? Yeah, it would drop. It would drop a bit early, yeah? Because it's nearly there. You add a bit of energy to the system, and the drop falls. What would happen if you flicked the tap at any other point in the cycle? When it's just drip maybe and water's just building up. Would you get a drip straight away? So could you get two drips together? Let's just go. What's the best way to do this? Yeah, here we go. If you flicked it there, when the water's just gone and there's only just the start of a drip coming, are you going to get another drip? The answer is no, you wouldn't. The energy that you add to the system only gets a chance to influence the system just before the system was set to go off anyway. What do you think would happen to the wavelength of the next oscillation after you flick the tap and the drop goes early? In flicking this at the end of one cycle, you're not altering the rate of water flow in the next cycle. Okay? So what we've got here is something that's stable. You can only influence its wavelength at certain points in its duty cycle, and when you do influence its wavelength at those duty cycle points, you only influence it for that wavelength. It resets itself and it carries on ticking just the same. So this is an inherently stable system that is stable to external inputs. And we've seen this kind of system before. Everybody remember this hippocampal cell? Yeah? And we said that this hippocampal cell was firing bursts of action potentials with spaces between them. Can you see how that's a burst oscillator? Yep, it's oscillating. Drip, pause. Drip, pause. Drip, pause. Yep. And if you were to change the effect at each of these, then you're still going to get that same oscillation. You might change the wavelength by other measures, but you have to make a profound change. You have to take away probably a half of its dendritic processes to start to change the frequency. And it's still oscillating. What's really interesting about this is that for a typical cell under typical function, the duration of this wavelength, or the distance between the oscillations, is about the length of time that an excitatory postsynaptic potential or an inhibitory postsynaptic potential can have its effect on a cell membrane. So what we've got is, we've got information coming into this cell from all over the place during this period and altering those membrane potentials and dendritic processes through inhibitory and excitatory postsynaptic potentials. But the cell only becomes unstable and able to generate action potentials at the end of its cycle. So all the information's coming in, cell decides what to do about it and does something and resets itself. And now the next lot of information is coming in, and the cell resets itself. Okay? So information is coming in from lots of different places. And if you think about it, information from different places takes different amounts of time to get there. Because of the limitation, I suppose, of the nerve conduction velocity in the axons. The longer the axon, the longer it takes for the information to get there. So the cell's receiving information from the cells adjacent to it, information from the cells a bit further away, information from all the cells miles away, putting it all together and generating an output, which is going to other cells, and then the next cells are coming in. So the whole thing is working with a kind of a tension relaxation. Processing information and output, waiting for the next round of input. Processing and waiting. It's a very similar system to the clock system in a computer that regulates all of the processes and makes them all think at the same speed at the same time. So when we look at these frequencies that the brain generates, what do we see? This is a power spectrum of a typical electroencephalogram. So this is activity recorded from the surface of the scalp in a human. And instead of being illustrated as changes in voltage over time, we've done some maths to it, and we've illustrated it as changes in power over the natural logarithm of its frequency. And so that you can see, or you can see rather, that there are changes in the energy in this waveform at different frequencies. And you can see a number of peaks within that. And those peaks correspond with some of the major oscillators within the central nervous system. But the whole electroencephalogram itself looks very complicated. What I want to do now is to break the EEG signal down and talk about how we can get this complicated process, this complicated waveform, from a number of relatively much more simple processes. If you've got a single biological process, what can you say usually about its frequency distribution? There's something that so many biological functions show this that often we don't test for it when we're doing stats. We just assume it's the case. We probably should test for it. Okay, who's seen one of those? What is that? Normal distribution. What are most biological processes? Normally distributed. Good. Okay. And a normal distribution has a very simple frequency distribution curve. Yeah? And we can say all sorts of interesting statistical things about that curve because we know it in such intimate detail. This looks more complicated, but you can kind of see on a frequency scan that it might well be the summation of a number of normally distributed curves. Can people see a number of normally distributed curves kind of embedded into that? Yep. So we've still got a relatively straightforward set of generators. How can they lead to the complexity of the EEG signal? Well, I want to use the analogy of a vase for this, because if you do certain things to a vase, and a vase that's as pretty and pleasant as that probably should have this done to it all the time, it looks very much like the EEG in terms of processes. If you take this vase and drop it on the floor, what's going to happen to it? It's going to shatter into a million pieces, depending on how hard you drop it on the floor. And if it was me and I owned that vase, I'd probably drop it very hard on the floor. Okay? What can you say about the size of the pieces? They vary, yeah? And they vary in a specific way. They vary in a way that fits into a number of normal distributions. So here's a very simple model of what happens when you drop a vase on the floor. It shatters around the point where it hits the floor, or a point where the energy focuses in the vase when it hits the floor, and a number of lines spread out across the surface of the vase, and it falls into those sort of segments. Is that what it looks like? Yeah? Do we get that sort of simple shape appearance when we drop a vase on the floor? It looks a bit more complicated than that, doesn't it? What I'm going to do now is I'm going to add in a number of focuses of energy on the surface of the vase, and I'm going to distribute them at random. They're all going to look just like this, with a dot and these 6 lines coming out from it. So they're all going to be relatively simple processes. Those are the random points that I just sort of placed over the surface of the vase as focuses of fractures. And there are the pieces that you would get with those fractures all working in the same way. Now, have we got the same regularity of piece size there? Or have we got lots of different pieces of lots of different sizes? Does that look like more like the kind of thing that you would expect to find on the floor after you dropped a vase? Yeah? Lots of different pieces, lots of different sizes. But it's all built out of this simple repeating process with a little bit of randomness thrown in. Okay? And that's how the EEG works. It's a very complex, in fact we call it pseudo-random, which means that, hey, we know it's not random because it's generated by these logical processes, but it's so complicated that when we're analysing it, we have to treat it like it's a random waveform because we just can't make any sense of it otherwise. So it's a pseudo-random waveform, but it's grown out of the summation of these much more simplistic processes, different oscillators. And if we look at the different oscillators in the human brain, these are the kind of oscillations that we get. And we're beginning to think now that each of these oscillations is, 1, a normally distributed process, and 2, originates from a different place in the central nervous system. And the complex function that we get from the central nervous system is an emergent function that grows out of the complexity that we get when we add all these things together. So it's not in itself very complex. It just looks complex because so much simplicity has been added together that the complexity emerges out of it. We've got all this regularity going on throughout the central nervous system, but what we see is that little bit of complexity around the edges because that's what's interesting to us. And so we've got things like 10 to 30, the beta waveforms and the alpha waveforms, which seem to control much of the function of the cerebral cortices. And down here we've got the much slower waveforms that seem to have a lot more to do with the hypothalamus and function in the spinal cord. And right up at the top we've got some fast and ultra-fast waveforms which appear when we're really focused in thinking on something and really concentrating very hard. So we've got different frequencies, different waveforms, all working together to generate that overall function. And it's really complicated, so I want to show you now a much simpler set of oscillations that lead to function to give you an indication of how these straightforward processes can begin to be useful. These are sections through a lamprey, a very, very simplistic, but nonetheless vertebrate, chordate at least, fishy thing, which has a descending reticular formation. This is a cranial slice, and this is a more caudal slice. And at each segment of the body, it has a motor outflow, which goes to the musculature. And the motor outflow is controlled, A, from this descending information, which progressively moves down the animal, but also from these excitatory and inhibitory networks, which pass the information from one side to the other. So that excitation on this side excites the muscles on this side and they contract, but it inhibits the muscles on this side. And excitation on this side inhibits the muscles on this side and excites the muscles on this side, so that we tend to get an oscillatory contraction. If you look at this diagram of a lamprey with 4 sensing electrodes in it, we're looking at the EMG activity, the electromyographic activity at those 4 sites, and you can see those 2 concepts. If you look at 1 and 3, which are opposite each other at the same level, you can see that they are mutually inhibitory, so that when 1 is contracting, 3 is silent. And when 3 contracts, 1 is silent. And 1 and silence, and 3 and silence. They're opposite each other. And 2 and 4 are also inhibitory to each other. 2 and nothing, 4 and nothing. 2 and nothing, 4 and nothing. But if we look at 3 and 4, you can see that 4, which is caudal to 3, kind of follows 3 each time. So you can get this concept that this activity is moving caudally down the fish's body. And if you think about how that caudally moving inhibitory information affects the movement of the fish, what you develop is a swimming motion. So that as the animal is swimming, these contracted areas of muscle move down the animal, and then you get another contraction on the opposite side of the animal, which also moves down the animal, and the animal kind of wriggles. There it is in a very simple and straightforward fish. Here it is in a much more complex fish. But can you still see how we've got that inhibitory moving, sort of wriggling thing moving down the animal's body, which is giving it motor function? And now all the animal has to do in order to swim forwards is to have a centre in the brain which says, let's move forwards, swim a bit faster. Let's not, swim a bit slower. And the spinal cord is looking after the ins and outs of that movement for itself and doing very nicely. And in fact, that's the case even up to the mammals. So if you look at a mammal like me and I'm walking, you'll tend to find that when I'm walking comfortably, I've got opposing swinging movement in my four limbs. When this arm's going forwards, this leg's going forwards, and when this arm's going forwards, this leg's going forwards, yeah? Now, I can pace if I try and think about it, so I can override that and I can do something different, but I've got to think about that. I've really got to sort of put some effort into it, and as soon as I stop thinking about it and go back to the lecture, I'm back into that swinging gait because that's natural for me. It's all about the spinal cord looking after the moving so that my central nervous system can do other things. And in fact, there is a motor centre, the mesencephalic locomotor centre in my midbrain, which is telling this system how fast to walk. And if you look at most mammals, the spinal cord will adjust the gait of the animal based on how fast the mesencephalic locomotor centre is telling it to walk. So that I don't gallop very well. But if you look at a horse or a cat, then it will have this swinging gait to start with, but as soon as it starts to go fast enough, it will move into this galloping gait. And it's the spinal cord that controls that gait. And then there will be a centre higher up than the mesencephalic locomotor centre, which is adding, or which is functioning at a higher centre. So for me, there might be a centre in my hippocampus saying, go to the end of the room, turn right, go along the corridor. Yeah? And even higher up in the volitional centres, there is a centre which is just saying, let's go to the pub. Okay? Doesn't have to worry about walking, doesn't have to worry about the route, just worries about what it wants to be doing. And so just as the sensory system is building up in terms of complexity, the motor system is building down in terms of volitional complexity. And these oscillations, which are building up in the motor system, are all a part of that. Talked about a few neural networks within the central nervous system. We've talked about the hippocampus. We've talked a little bit about the cerebellum and about the cerebral cortex, and I'll be talking about those in more detail next lecture. But I just want to introduce them now to talk about how structure serves function. So different networks have different jobs to do. And different networks have different structures. And the different structures within the different parts of the central nervous system make them suited towards different jobs. They have different constraints and restrictions on their function, which is imposed by their structure. They have different needs and requirements, particularly in terms of the energy that it takes to run them, based on their structure. But the structure serves the function. So if you look at something that's doing something, it has a structure which makes it quite efficient at doing that. And similarly, if you look at the structure of a neural network, you can make a guess at what it's probably doing based on what it looks like. So I'm just going to talk about three different classifications of structure now. This is a small world network. And what it means is that each neuron is very likely to be connected to its near neighbours and probably the neighbours next door but one as well. But there are no connections in distance over this. So this neuron is not going to be connected directly to this neuron. And if this neuron wants to speak to this neuron, it's got to speak to that one, and speak to that one, and speak to that one, and speak to that one. Okay, so there's a lot of passing information along in this network, a lot of Chinese whispers. But it's very good at processing information quickly. Because if this neuron gets information, it's only got to pass it next door or next door but one, and it's all very quick and very rapid. So it can really only process simple information about particular areas of the body maybe, but it can do that very, very quickly. And we'll see in the next lecture that that's what the cerebellum looks like. And the cerebellum, surprise, surprise, is concerned with coordination of motor function throughout the body, got to be done quickly, but pretty much this hand doesn't really care what this hand is doing. If this hand starts to do this, what does this hand care? It's happy. So this part of the cerebellum doesn't need to speak to this part of the cerebellum. This small world network works really well. The next network up is the random network. In this network, the random network, cells are just as likely to be connected to cells far away as they are to be connected to their near neighbours. Okay? Every cell is randomly connected to every other cell. The hippocampus tends to look like this. The hippocampus is a flexible thinking space within the forebrain, and flexibly deals with things like short-term memory, spatial orientation, and the space in the room that we're in, that kind of thing. And it has this randomness of connection. It's good at dealing with global information, global processing, but the axons are longer in this system than they are in the small world network, and so information processing is necessarily a little bit slower. And if you were to measure the overall length of axon in this system compared to the small world, you'd find that there's a lot more axon in this system per neuron than in the small world system. So it's a less efficient system. It uses more energy to make that system. So we've looked at the small world network. We've looked at the random network. If we look at a network in between, we've got an intermediate network. You can see that this has got elements of the small world system in it, but it's also got a few longer connections. And so what it's doing is it's saying, if this neuron wants to speak to this neuron, it doesn't have to go all the way around the edge. It can go to its nearest long-term connection, hitch a ride, and then get there. It's a bit like flying by air. You take a flight from Palmerston North to Auckland to get to the big airport, and then you go anywhere you like, and then when you get to where you're going, you take another short flight to get to the airport nearest to the place you actually want to go to. And this has features of the small world network. It can deal with information relatively quickly, and features of the random network. It can talk across the network relatively well. And it's also intermediate in terms of its energy needs between the small world system and the random system. And the cerebral cortex looks like that. So we've got three cortices there, the cerebellum, the hippocampus, and the cerebral cortex. Three different architectures and three different functions, three different sets of advantages and disadvantages. Or rather, two and one that's intermediate. So what do oscillations do in neural networks? Well, as I said earlier in the lecture, they provide functional binding. They bind the cells together in terms of how they work together, in terms of the temporal nature of their function. And different functions need different binding. Therefore, different structures need different binding. Therefore, we need lots of different oscillatory patterns throughout the central nervous system to serve those different structures, to provide those different functions. So, hopefully from that, you can put all that together and begin to understand how this cell in the hippocampus is providing its function by oscillating at these different frequencies, and how in doing so it's not driving its own oscillations, but it's being driven by a much bigger system which is coordinating everything together. And that system is being driven by lots of different oscillations. We know the origin of some of these oscillations, but we don't know the origin of all of them. And we've found these different oscillations by deconstructing the frequency spectrum of the electroencephalogram rather than by finding different structures within the brain that oscillate at those frequencies. We know that some of them come from particular places. For example, the alpha wave, which operates at around about 20 hertz, we know comes from an interaction between the thalamus and the cortex. We call it a thalamocortical waveform. We know that there are much faster waveforms which are generated by an interaction in the pons and the cerebellum. Again, those faster waveforms are supporting that different structure. We know that there are oscillations which run between different parts of the cerebral cortex, sometimes through the corpus callosum, and alter activity within the cerebral cortex. And in terms of the brainstem, the periaqueductal grey seems to be an area that tends to generate oscillations and controls, in inverted commas, much of that brainstem. Interestingly, going back to where we started, the rhythms in music seem to reflect the rhythms in brain function. So when we are clapping at particular rhythms, and the rhythms that we clap at the best seem to be between about half a hertz and about 3 hertz, we're reflecting particular rhythms in the central nervous system in this kind of area, which are generated by those oscillations. So that rhythmicity in music is a reflection of the rhythmicity inside our central nervous systems. And it's it makes us feel good and it makes us feel happy and calm because it's reflecting what's going on inside us and we can kind of relate to it.